Good morning. Today we discuss about the cholesterol metabolism. Cholesterol. This is the another most biological compound which is essential for our body. But sometimes it is excess which is fatal in our body. The cholesterol, the word cholesterol is derived from the Greek word chole means bile and steros means solid and OL equal to alcohol. That means solid alcohol. And cholesterol is widely distributed into the body. In a 70 grams man, a total of about 140 grams of cholesterol is available, which is distributed as about 30 grams in the brain and nerves 30 grams, and in the muscle 30 grams, in the adipose 20 grams, and this 10 gram in the skin and 10 gram in the liver and spleen, 5 grams in the bone marrow and 3 grams in the alimentary tracts and 2 grams of the adrenal gland. Cholesterol is the compound which is essential in your body, and every day per day 1 gram of cholesterol will be synthesized in your body, and which is essential for the synthesis of the all the steroid hormones, all the vitamins, vitamin D and bile acids. So many compound is synthesized from the cholesterol. So cholesterol is the precursor molecule for all the above preparation, but if it is high, just cross the limit, just 200 mg per dl, just cross the limit, it will produce sometimes heart disease, and that's why the cholesterol is sometimes it is good and sometimes it is bad. It is called bad cholesterol and good cholesterol. So another my request for the situations, we can stay home and stay safe for prevent COVID-19. It is the burning situation in India, not only the India, whole over the country. The coronavirus spread throughout the world, and every day so many people are affected by this virus. It can be prevented by the social distancing and by stay in the home. So please, I again request you to stay at your home for prevent the COVID-19. Let us start the metabolism of cholesterol. First, cholesterol, which is light yellow crystalline solids soluble in the chloroform, because it is a lipid in nature which is solubilized in the fat soluble, and the basic structure of the cholesterol which is cyclopentanoparhydro and distributed in the brain, nervous, muscles, adipose tissues, skin, blood, liver and spleen. It is totally absent in the plant. This is the basic structure of the cholesterol. Basically it contains 27 carbon atom. Basically this ABCD ring, you see this is the basic steroidal nucleus. The name is the cyclopentanoparhydrophenanthrin ring, or sometimes parhydrocyclopentanophenoxyphenanthrin rings. This ABCD, the basic rings of cyclopentano, all these steroid, not only this steroid, cholesterol also contain another sub carbon atom, total 19 carbon atom, but here it is the 27 carbon atom consist of cholesterol. This is the structure of cholesterol. The structure, it has a cyclopentanoparhydrophenanthrin already discussed. It has a 27 carbon atom and one hydroxyl group in the third position, and the double bond present between the 5 and 6 atoms and 8 in the side chain. What the function? The function of cholesterol is very important. There are so many functions. First of all, the cholesterol is the precursor for the synthesis of vitamin D as well as the bile acid. Vitamin D, which is essential for the prevent osteomalacia and rickets, and bile acid, which is essential for the lipid absorption, and cell membrane, which is another component, the permeability, which is modulating effect on the fluid state, fluid mosaic model of the cell membrane, which is can be controlled by the cholesterol, and nerve conduction, it is also controlled by the nerve fibers by the cholesterol, and fatty acid transport, which is by the cholesterol ester for the oxidation, and steroid hormone like glucocorticoids, androgen, estrogen, progesterone, all are synthesized from the cholesterol. These are the precursors molecule of cholesterol and essential ingredient in the structure of lipoprotein. This is the another function of cholesterol. Now let us start the synthesis of cholesterol. How to synthesize? Basically it is synthesized in the liver. All other compound, just like a compound which is synthesized in the liver, basically which is coming from the diet, or it will be synthesized from the liver. The basically it will be synthesized as like a ketone body. Here is a 2-acetyl CoA, or that means acetyl CoA, which is coming from the either fats or from the carbohydrate. It will be this acetyl CoA, 2-acetyl CoA in presence of acetyl CoA synthase, it converted into the acetyl CoA. The next step, another acetyl CoA which is incorporated into the this acetyl CoA by the presence of HMG CoA synthase, which is the key factors or regulating factors, which is formation of beta hydroxy beta methyl glutaraldehyde CoA. In the presence of NADPH and NADH, another HMG CoA reductase, which is for formation of a mevalonate CoA. It is conversion of 6 carbon atoms, and these produce 3-phospho 5-pyrophospho mevalonate. Another next steps is the decarboxylation. It is production of the decarboxylase 3-phospho 5-phosphopyrrolate decarboxylase, which formation of isopentyl pyrophosphate. This isopentyl pyrophosphate and pyrophosphate, which is the geranyl pyrophosphate. And the next transferase enzymes isopropyl for the farnesyl pyrophosphate. It formation of farnesyl pyrophosphate to squalene. Next steps in the steps 6, it is by the presence of cyclase, it forms a lanosterol. This lanosterol 30C, which is again it is reductase enzyme gymosterol, and ultimately desmosterol, and finally cholesterol will become the liver. So there is so many reaction, and in this case there is a Rodenbach and John confirmed, which are get Nobel Prize in the 1975 for the biosynthesis of the cholesterol. So this is the pathway by which cholesterol will be synthesized. Now the regulation of cholesterol biosynthesis, how it will be regularized. Here the two pictures are given from the two books for the betterment of the student, for better understanding. See these books, first one is the Vasudevan and second one is the U. Satyanarayana. The first is, but basic thing is the same. First, how to regulate. They are the key step. HMG CoA reductase, the enzyme which is the key factor, key regulator for the synthesis of cholesterol. These enzymes, all enzymes which are protein in nature, and every protein which is synthesized in control under the genes. The first of the gene, which is first gene, a particular gene which transcription, after the translation, the particular protein or enzymes like HMG CoA or hydroxymethyl CoA reductase, which is the influence of the particular gene. This gene is reduced or inhibited by the directly cholesterol. If the HMG CoA is activated, in that case cholesterol can directly it inhibits. But apart from this HMG CoA also inhibited by the so many hormone also. So many hormone can inhibit this. First of all, the glucagon, corticosteroid, at which is reduce the HMG CoA reductase, and some also enzymes which is inhibition. Some statin group of drugs like simvastatin, pravastatin, all are the drugs which are inhibit the HMG CoA reductase. Another which is activation of the HMG CoA reductase, and which is essential for the synthesis of cholesterol. The main hormone which is influence the HMG CoA reductase, insulin, thyroxine. There is the two dephosphorylated form which is active, insulin and thyroxine, two hormone which is accelerate the synthesis of cholesterol by the influence of enzymes HMG CoA reductase. The formation of HMG CoA to mevalonate, finally it will be produce the cholesterol. In case of there is some transcription factors in this. The second picture, the same HMG CoA reductase, which is the first is the DNA to a transcription factor, which is produce some mRNA. This particular mRNA produce the enzyme protein HMG CoA reductase. The reductase, which is inhibited by the insulin, and glucagon, which is produced here by the same things we discuss in the two picture. Then excretion of cholesterol. How to, if the excess cholesterol will be excreted through the bile properly, esterification with the PUFA or polyunsaturated fatty acid, and partly reabsorbed from the intestine. Unabsorbed portion is acted by the intestinal bacteria to form a cholesterol and coprostenol, which is excreted as a fecal sterol. Another part is converted into the bile acid and excreted in the bile salt. Next, the degradation of cholesterol or the synthesis of bile acid. How to synthesize? The cholesterol is the precursor molecule which is synthesized in the liver. First, the cholesterol and 7-dehydroxylase, alpha hydroxylase. Then the several step of reaction, it is formation of cholic acid, and cholic acid with glycine, glycocholic acid, and in case of taurine, taurocholic acid. In the intestinal bacteria, it formation deoxycholic acid. Another several steps from the starting from the 7-hydroxycholesterol, it conveys chino-deoxycholic acid. Then another molecule taurine or glycine in the tauro or glycocholic acid is formed, and with the intestinal bacteria which is from the lithocholic acid. So there is the formation of the bile acid for the degradation of cholesterol, and bile acid which is essential for the absorption or emulsification of the lipid. So that means the degraded product of cholesterol which is utilized for the absorption of lipid, which is essential for the bile acid. This is for your exam purpose, which will be very essential, how to formation of bile acids. Next, synthesis of steroid hormone. So many steroid hormones, all the steroid hormone which are synthesized from the cholesterol, because cholesterol is the compound which is synthesized all these steroid hormone, and this steroid hormone is essential for our body for different biochemical reaction like estrogen, progesterone, androgen, and so many hormones. The cholesterol 27C, it forms a first pregnenolone into progesterone, and progesterone is a cortisone which is anti-inflammatory reactor, and progesterone which is control the so many reproductive system, aldosterone which is adrenocorticotropic hormone, and estradiol which is another estrogenic hormone which is control the pregnancy to another reproductive system also. All these hormones, steroidal, basically all steroid hormones, all are synthesis from the cholesterol. Now the synthesis of vitamin D, which is essential for the development of the bone. The cholesterol is the main precursor for the synthesis of the vitamin D for prevention of the osteoporosis, rickettsia, and so many disorders. The cholesterol fat is converted into 7-dehydrocholesterol in presence of UV light by skin. The cholecalciferol which is formed, cholecalciferol with the vitamin D3 in the liver, 25 cholecalciferol is formed in the kidney, or parathyroid hormone is 125 dihydrocholecalciferol or vitamin D2 which will be formed. There are two types of vitamin D2 and D3 which is formed. In the precursor molecule is the cholesterol, basically in the presence of ultraviolet ray, and which is occur first in the liver, then in the kidney, and basically occur in the skin. So thank you for this patience to listening this lecture, and thank you all. If you like it, please subscribe it for my channel. Thank you all the students for listening.